Thank you very much, Andy, for your kind introduction. And I also would like to take this opportunity to thank our organizers to invite me to here. And as Andy said, my colleague, Dr. Tian Wang, couldn't make it. He was supposed to talk about the synaptic of the ketamine, and I'm sort of like a substitute. And very fortunately, although my lab is very much focused on Alzheimer's disease, and we are looking for the dementia, and the ketamine was one of the drug could be induce the dementia. So one of my project is working on the ketamine's effect. And as you know, so I changed the title. I'm not much talking about the synaptic plasticity, but more about the ketamine, the good and the bad, and in the mental health. So as you know, the ketamine is a very, very popular and anesthetic, and also recently is a very popular club drugs and illicit drugs in particularly in the North America. And this drug and also being used in lots of the pregnant women and in the childbearing ages in the women. And so more recently, as psychiatrist knows, and ketamine attracts extensive interest because it's very quick and effective for antidepressant property, and also in many of the trials and showing that ketamine could be really good in the antidepressant in the urgent conditions, and also combined with the ECTs. But unfortunately, the ketamine, as I said, is also the very most common illicit drug. They could induce lots of the dissociative and also side effect, and particularly to the women. And this was very popular in the childbearing ages. So it could have some of the very unforeseen side effect. And one of our interesting is this going to affect the child development, or particularly in the embryonic stage when the women at the pregnancy taking this drug as either as an antidepressant or as club drugs. So we want to know if ketamine could induce lots of the side effects, and this would be something we have to be careful if we are using this drug as an antidepressant to the patient, particularly if they have the period to bearing the kids or during the pregnancy. So, however, the ketamine's effect on the embryonic development has not really been well studied. So we took this subject, and we want to know if this has anything to do with embryonic development. One of the major things in the neurodevelopment is neurocrest development. As you know, the neurocrest is a vertebrate specific protruent cells population derived from the embryonic ectodermal cell layers, and it is derived from border regions between the epidermis and the neuroplate. And the neurocrest could give rise to numerous derivatives, including the cranial cartilage, neurons, glial, and of the peripheral nervous system and heart outflow tracts. So abnormal development of the neurocrest derivative could result in the neurocrestopathies, leading to many human congenital disorders such as HD and Conning syndrome, Watson's syndromes, DiGeorge syndromes, and Charge syndromes, and also neuroblastoma and melanomas. So, as we know, the regulation of the neurocrest is very tightly controlled and regulated. And many of the signaling pathway we have known, like the Wnt signaling pathway, Notch signaling pathway, retinoic acid pathways, signaling pathway, has played major role in control the how neurocrest in development, and how the neurocrest differentiate into the different organs and the different cell types. So we would like to know if this ketamine applied to the early development stage could affect the neurocrest development. One of the model system we use is the Synaptic system. As you know, there is two kind of the Synaptic. One is Synaptic larvus and Synaptic tropicanus. So the Synaptic system is very good for the development study because they have a very short lifespans and range from the 4 months to 1 to 2 years, depending on the different species. So if we apply the ketamines, and then we could quickly know what kind of the neurodevelopment or in general, the development has been affected. So what we did is that we treated this Synapse at a very early X stages, and showing us that when we treated this ketamines and the axons, soul mass, and also the pigmentations of those synapses has been changed, altered. There is a significant inhibition of those megacolon-like the phenotype and also cranial cartilage atrophy. So this is very much reflective of the neurocrest development abnormalities. So then we went on to show what kind of the genes in the neurocrest inductions has been affected by treating the ketamines. What we found is that actually the ketamines blocks neurocrest induction independent of mesodome genes. So what we can see is the SNAG, which is one of the neurocrest development genes, has been inhibited if we applied those of the ketamines. However, the mesodome genes, they're reflected by this several of the gene markers, like CORA, 8 and the FIGA, and none of them has been affected. Indicate of the neurocrest induction has been affected independent of the mesodome genes. So then we went on, what is specifically of the neurocrest induction has been affected? Turns out, if we look at all those genes involved in the neurocrest, the ZIC5 pop up as one of the major genes has been affected by the ketamines. So if we apply the ketamine to those synapses, and what we found is that the ZIC5 has been significantly reduced. And certainly this is depend on the different stage of the embryonic development, and you certainly see the ZIC5 has been reduced. So this is very interesting. So we identified the ZIC5 gene has been affected by this ketamine treatment. That certainly is affected the neurocrest development. So then we want to see if ZIC5, if we do the rescue experiment, we introduce that the ZIC5 mRNA, we can inject it into this ketamine-treated synapse. What we can see is that actually they can restore of those SNAG, which reflect the neurocrest induction stages. So the SNAG gene has been restored. So the ZIC5 affected by the ketamine and then downstreamly affected of the SNAG, which essentially affects the neurocrest inductions. So then we understand, so the ZIC5 and the SNAG has been regulated previously by the Notch signaling pathway. We know the Notch signaling pathway is extremely important neuronal transduction molecules, and it really controls one of the very important phenotype of the neurodevelopment called the lateral inhibition. So if you have one progenitor cell has a notch being activated, so surrounding the neuroprogenitor cells will not become the neuron because of a lateral inhibition. And the previous work from my lab and others has been showing the presenilin, which one was very much associated with Alzheimer's disease development, is required for control the notch signaling. So we discovered and that presenilin knockout will block the notch signaling pathway, and that will lead into this very typical notch phenotype. So with the somatic shortens and also cerebral hemorrhage and also cavity enlarged. That's very typical of the phenotype of the notch deficiency. So then we went on to say, because we already see the ZIC5 and then SNAG has been affected by the ketamines, indicate one of the possible signaling pathways is notch controlled. So we wanted to know if that the notch signaling has been affected by this ketamines, that in turn affect the ZIC5 and then SNAG. So yes, lo and behold, what we found is that the Notch target gene, very typical Hess genes, Hess 5.1, B and A, two isoforms, has been really reduced or inhibited by the ketamine treatment. So, and furthermore, and what we found is that all those ketamine inhibition induced very much Notch deficiency phenotype in this xenopus, with this body axis being shortened, and also shows those of the vagalescent structures, very typical of the Notch phenotype. So then we want to identify what's exactly the Notch signaling being affected by this ketamine. Where is the molecule being affected, or in what kind of the molecular levels? So then we went on to look at this ketamines and see if the Notch protein level has been affected. Yes, we found the Notch, actually ketamine affected the Notch homeostasis. The Notch total protein level has been affected. Furthermore, we want to know if this ketamine regulated the Notch the degradation or Notch's transcription. And then we cloned one of the piece of the xenopus Notch target genes promoters. Then we identify a couple of the regions that it's ZIC5's promoter genes regions. It's really regulated containing of the Notch response element. So if we applied this ketamines, so then we'll certainly reduce of the promoter activity of ZIC5, which is containing the Notch response element. So that's absolutely demonstrated this Notch phenotype is caused by the ketamine effect on the ZIC5's Notch response element. And furthermore, we want to do some of a rescue experiment to show if this is Notch signaling affects the ZIC5 and the SNRC gene can be restored by the Notch inductions. So it turned out, yes, what we found is that either we use that the injection at the 4-cell stage, very early stage of xenopus embryonic development, we injected of the Notch activators, and it turned out that and then we certainly can rescue of this ZIC5's expressions. So if on this side we don't inject of the activators, so the ZIC5 genes has been blocked or inhibited. But if we inject the Notch NSAID, and that's the activator of the Notch signaling, then we can restore this ZIC5's expression, which is reflected the neurocrystal development restorations. So in summary, what we found is that the exposure of the ketamine, one of the recent popular antidepressants and also anesthetics, and also the club drugs, is really has the very fundamental harmful effect on the embryonic development. So they could affect in the animal model system, in the early neurocrystal developments. And what we identify the molecular pathways underlying this ketamine's effect on the neurodevelopment. And furthermore, we found that the Notch is really the one played a major role, mediator of the ketamine's effect on the embryonic development. So furthermore, and we identify one of the ZIC genes promoter region containing of this 15 base pair co-binding element that's mediated of ketamine's effect on the ZIC5 gene expressions via this Notch signaling pathways. So in turn, very simple cartoons to show in the normal conditions, so the Notch signaling will trigger off ZIC5 and induce the neurocrystal inductions. However, if you have the abnormal use of the ketamines, so that could affect the degradation of the ICD, the Notch activators, and further reduce of this ZIC5's gene expression through its transcription and affect of the neurocrystal development. So what we want to say is that ZIC5 work as a downstream of the target of the Notch signaling in neurocrystal induction, and ketamine disrupted Notch neurocrystal inductions will Notch ZIC5 signaling pathway on novel pathways underlying ketamine's effect on the early embryonic development. So this result was published last year. And this is most done by one of my former postdoc, and now it's associate professor in the Children's Hospital, Dr. Yu Si in the Chongqing, and also with the collaboration of Chinese Academy and the Kunming